So the mycoplasma pneumoniae. General features: these are the smallest free living organism. Smallest free living organism known to the environment, and it is like the viruses. Why it is like the viruses? Because of the two properties. First, it is the smaller in size, and that's why it can pass through the bacterial filter. Pass through the bacterial filter. It lacks the rigid cell wall, but the cell membrane contain the sterols. Cell membrane contains the sterols. It is completely resistant to beta-lactam antibiotics acting on the cell wall. It shows the gliding motility. It shows the gliding motility because of the specialized hyphal structures at the tip of organism. Then it is pleomorphic in nature. Pleomorphic in nature. It is not very well stained by the Gram stain, but well stained by the Giemsa stain and other pathological stains. Then Mycoplasma pneumoniae and L forms. L forms are particularly important. So L forms, they usually L forms they are the cell wall deficient form. Cell wall deficient forms, either because of the beta-lactam antibiotics, or they lose their cell wall spontaneously. But the Mycoplasma pneumoniae has the completely lack of the cell wall, so it is having the complete and stable L form. Then it is also known as PPLO agent, pleuropneumonia like organism, or Eaton's agent, or the PPLO agent. Myco means fungus like branching filament. Fungus like branching filaments. And plasma means plasticity of the shape. Plasticity of the shape. So it is known as the mycoplasma. After studying the general features of the mycoplasma, let us move toward the antigenic structure of the mycoplasma. So there are the two parts: glycolipid antigen and cytoadhesin E1 molecule. Cytoadhesin E1 molecule. So glycolipid antigen responsible for the cold agglutination test. But the cytoadhesin P1 molecule responsible for the adhesion or attachment to the respiratory tract mucosa. Now, after attaching to the respiratory tract mucosa, it will cause damage to respiratory mucosa by either three things. Either it is because of the H2O2 hydrogen peroxide, or it is because of the cytolysine, or either it is because of the hydrogen peroxide or the cytolysine or the lipoproteins. So it is actually cytotoxin, vacuolating cyto. It is similar to the vacuolating cytotoxins, similar to the pertussis toxin. Lipoproteins, they produce increase in the inflammation. for the affected mucosa. Now coming to the immunity or immunopathogenesis of the mycoplasma, there are the two parts: cellular immunity as well as the humoral immunity. Cellular immunity actually exacerbate the pathogenesis of the pneumonia, so it is increasing the pneumonia. But humoral immunity that will help to prevent the disseminated mycoplasma infections, like that will prevent disseminated mycoplasma infection, particularly in the child, like arthritis or other joint infections or skin infections. Now moving toward the clinical feature, but first of all we will say it is having incubation period roughly around the 2 to 4 weeks, and it is transmitted by the aerosol as well as the droplets from the person to person. So school, military brackets, troops, or the daycare or the summer camps are having increased chance of the infection. Now in the clinical features of the mycoplasma pneumoniae, only 3 features. The first one is your URTI, the second one is your atypical pneumonia, and the third one is the other extrapulmonary manifestation. So URTI, first of all, URTI is having the most common manifestation of mycoplasma pneumoniae, including the pharyngitis. Upper respiratory tract infection is the most common manifestation of the mycoplasma pneumoniae. The second was atypical pneumonia. Atypical pneumonia, it is the one of the common agent for primary atypical community acquired atypical or interstitial pneumonia. So what will happen to the patient? Patient come with the fever plus or minus sore throat plus chills, rigors, etc. Plus there will be a non-productive cough in the atypical pneumonia. Chest X-ray, definitely we are having the interstitial infiltrates. And the third one was extrapulmonary manifestations, either because of the acute infection, it will manifest as the GBS, Guillain-Barré syndrome, or it is in the neurological symptoms we are having encephalopathy, or the GBS, or the meningitis. In the skin we are having the Guillain-Barré syndrome. Other features like the heart is affected, then pericarditis or myocarditis will be present, anemia, coagulopathy, etc. These are the rare features of the atypical. Then coming to the lab diagnosis of the mycoplasma pneumonia, first of all, the sample collection. Sample will be your oropharyngeal swab, it is, or nasopharyngeal swab, then throat swab and nasopharyngeal swab. They are the most common samples. They should be collected, then it is transported into the following two media, either it is in the VTM or in the standard mycoplasma transport medium. In the content of standard mycoplasma transport mediums are penicillin plus gelatin plus fetal bovine serum. Fetal bovine serum. Then in the viral transport medium, but it is having the ampicillin plus cefotaxime antibiotics in it. Ampicillin plus cefotaxime antibiotics present inside this. Now, culture part of the mycoplasma pneumonia. So in the culture part, the first and foremost is you have to take 5 medias are there for the mycoplasma. All are standard. Solid medium, standard solid medium, standard liquid medium, then Hayflick modified medium, then biphasic or we can say diphasic medium or SP4 medium. In the standard solid medium, there will be a PPLO agar plus gelatin plus penicillin will be there. But in the standard liquid medium, phenol red indicator is important and PPLO broth is important. Diphasic medium is having the both property of standard solid as well as the standard liquid medium. Then in the Hayflick modified medium, there will be a heart infusion broth inoculated in it. And in the SP4 medium, there will be a fetal bovine serum, similar to the standard mycoplasma transport medium. Fetal bovine serum will be. Now, how will you detect the growth of the mycoplasma? First on the solid medium, then second on the liquid medium. First we are trying the growth of the solid medium. So in the solid medium, what happens that on the typical appearance of the mycoplasma colonies, these are the colonies which are embedded on the agar surface plus central granular opaque area. This is the central granular opaque area surrounded by the peripheral translucent growth, hazy, or you can say peripheral translucent growth will be there in the solid medium, standard solid medium. Then this appearance is known as the fried egg morphology, the fried egg appearance of the mycoplasma. So this is like the fried egg. Now the colonies are so small you cannot see by naked eye. Your eye will be strained to identify because of the smaller size, 200 micrometer roughly size of the colony for mycoplasma, and around 80 micrometer, 40 to 80 micrometer for the urea plasma. They can be seen with the hand lens. You have to visualize the culture plate with the hand lens or with the Dines staining. So what is happening in the Dines staining? That plate is flooded with alcoholic solution of the methylene blue. Then there will be either a royal blue color or there will be a green color. If you see the royal blue color, then it is definitely your Mycoplasma pneumoniae, but urea plasma will be having the green color. Then after the growth on liquid media, so in the growth of liquid media there will be turbidity plus phenol red is there. In this standard liquid medium, we told that there will be a phenol red indicator. So the phenol, because of the phenol red indicator, the color will change from red to yellow because of the glucose fermentation. This happens only and only with Mycoplasma, and that too with the Mycoplasma pneumonia, not with the urea plasma, not with the other Mycoplasma species. After this two growth of the Mycoplasma pneumoniae on the solid as well as the liquid medium, we are having some other test. So in this other test, only names are important: hemadsorption inhibition test, then hemolytic colony, hemadsorption inhibition test, hemolytic colonies are there, tetrazolium reduction test, tetrazolium reduction test. Then after there will be some antigenic test. So antigenic test, you can directly detect the cytoadhesin P1 molecule by ELISA. Then other antibody detection test. In the antibody detection test, IgA detection will be helpful for the adult, IgM for the children. This will be helping to detect the acute infection of the Mycoplasma. These two molecules are important. Then non-specific antibody test. Non-specific antibody test. In the non-specific antibody test, we are having first cold agglutination test. Cold agglutination test, and second thing is Streptococcus mg test. Streptococcus mg test. Cold agglutination test. It is the since the Mycoplasma contain the heterophile antigens that will produce with the agglutinate with the heterophile antibodies, and so the cold agglutination test, the glycolipid antigen. So the glycolipid antigen will be is important for the heterophile antigen, heterophile agglutination test, that is your cold agglutination test. Here what happens, that I antigen of O blood group of human will agglutinate with heterophile antigen of Mycoplasma. It's known as the cold agglutination test. Another is Streptococcus mg test. It will kills the suspension of Streptococcus mg, group F Streptococcus cell wall. Then comes to the treatment part. Definitely your macrolides, azithromycin. In the last, agents of the atypical pneumonia. The first agent is Mycoplasma pneumonia, atypical bacterial pneumonia, definitely. Then second is Chlamydophila pneumonia, and third is Legionella pneumophila. So this was the note taking lecture. note taking for postgraduate university examinations. Simply listen while working in